This is Becky again. In this video, we'll look more closely at particular parts of Genome Browser. First, let's zoom out a bit so we can see a bigger region. The highlighted part here is the piece of the insert that we actually sequenced in one of the two reactions for this plasmid. We've got an EcoR1 site here at the left end of this highlighted part, but we're not seeing the other EcoR1 site, which must be over to the right somewhere. Unless an insert is pretty small, we don't usually expect to be able to sequence the entire thing. If we zoom out again and then scroll over, we can find the other EcoR1 site. In post-Lab 8 and in Lab 9, you should be able to use both sequence files so that you can verify where the ends of the insert are. Here, we'll just assume that this next EcoR1 site is the other end. As before, we can right-click on it to get the coordinates. There aren't any BAMH1 sites in this insert, but if there were, we could get their coordinates the same way. Next, let's look at some of these annotated sequence features. I'm going to zoom out a bit more, just so we can see a wider variety of things. You'll notice that some of these features are different colors. If you'll look closely, you'll see that each has a small arrow pointing to the left or to the right. Those arrows are showing the direction of transcription. Not all of these features are actually protein coding. Verified protein coding genes, like these two, are red. This beige one encodes a small nucleolar RNA rather than a protein. The gray one is marked as a dubious open reading frame, unlikely to encode a functional protein. Basically, someone was looking at possible reading frames, found one with a start codon, some other codons, and then a stop, so they flagged it as a possible gene. Later experiments showed that it probably wasn't, so they leave it marked so that no one else will waste their time on it. The pink ones, which look a lot like the beige ones, which is a nuisance, are for putative protein coding genes or protein coding genes of unknown function. If we want to look further at one of these genes, we can right-click on it and choose View Details. This will give you a brief description of what's known about the function, as well as the DNA sequence. For more information, we can click on the gene name here at the top to be taken to a summary page. You'll notice that there are two names listed, the standard name and the systematic name, sometimes called the ORF name. The standard name is shorter and easier to remember and work with, so anytime we ask you for the name of a gene, that's what we're looking for, the standard name. There's a lot more you can do from this page, but that will wait for lab. Best of luck in your sequence analysis.